here we go. So there's various terms you can use. We often use connectional anatomy or white matter anatomy, but we mean the same thing. Now, in terms of studying the brain, you can look at the brain from the surface anatomy, which is what we did yesterday. You can slice through the brain and look at the sectional anatomy. You can visualize the connections and look at the connectional anatomy, or with the help of fMRI, as you've just seen in the previous talk, you can look at the functional activated areas. But there's really only two methods that allow you to study the connections of the brain. And the first one, already briefly mentioned, is post-mortem Klingler dissection. But by definition, that is a post-mortem method. So we can't apply that to the living human brain, but we now have tractography, white matter tractography available to us that allows us to do the dissections, they're called in vivo dissections, in the living human brain, and we can combine the two. Now, I'm going to talk you briefly through both methods in terms of what you need and how they're done. I want to alert you to this wonderful paper that came out in November last year in Brain Structure and Function, and it's a review on the literature of Klingler dissection. So in this paper, they looked at the common methods, how to do it, and they found that most studies use a 10% formaldehyde to preserve the brain. The freezing time can vary between 8 hours to 2 months, and the temperature can vary from -5 to -80. But what all these methods have in common is that you fixate the brain, you freeze the brain, and you thaw the brain. And you can repeat that cycle several iterations long. Now, what you see in the picture already is that this fiber dissection technique involves peeling away the cortex to show the white matter that lies underneath it. And I don't get scared, it's not a real brain, but we basically start from something that looks like this. It's half a hemisphere, so you see the size nicely, to something that looks like this. And you can already appreciate that this is a tricky endeavor because it's a super thin brain slice in the end. And it's fine here because this one is rubber, but the real brain is tricky to handle once it gets to that level where it's super thin. So how do you get there? How do you make that happen? Well, you need basically a couple of things only. You need a specimen, which is actually the hardest part of it. You need a lot of patience. So it takes about 2-3 days, depending on your skills, but you'll be there for a little while. You need instruments, but as you can see, nothing fancy. So the spatula, we usually make them ourselves to have the right shapes. The other materials are forceps, scissors, and a bowl of water and a syringe. And ideally, but not necessarily, a microscope, but it greatly helps with the dissections. And then you basically start from the surface of the brain. Now, just a little recap from yesterday. I specifically chose one of the examples where the anatomy is not as straightforward. But if we start with the main landmarks, so the Sylvian fissure, the lateral fissure that separates the frontal and the temporal lobe. Then we have the central sulcus that separates the frontal and the parietal lobe. Once you identify the central sulcus, you easily have the pre- and the post-central gyri. Then behind the post-central gyri and around the tip of the end of the Sylvian fissure, we have the supramarginal gyrus. Then the next, if we go down into the temporal lobe, we now can see the superior temporal sulcus that's nicely wiggling through the temporal lobe here. And on the tip of that, the posterior end of it, and just posterior to the supramarginal gyrus, you have the angular gyrus. The supramarginal and the angular together are the inferior parietal lobe. So we can already see that now we have an area here that is nicely delineated, which is the superior temporal gyrus. And then I just show you where the superior parietal lobe is as well. Now you can finish the brain in your own time. This is just to get you started and to make the point that we usually start our dissections from the superior temporal sulcus. And the way you do it is that you slowly peel away the cortex in the depth of the sulcus to expose the very superficial white matter fibers that are called the U-shaped fibers. Now, once you do that, you get a beautiful dissection like this one. So just to give you a bit of orientation, because I know I just took away the landmarks. So here's a rough lobar division, the frontal, the parietal, the occipital, and the temporal lobes. We still preserve the Sylvian fissure, but you will now struggle to identify where the central sulcus was. But what we exposed by peeling away the cortex are the U-shaped fibers connecting neighboring gyri, and you can nicely see the lovely fibers of the arcuate fasciculus that is arching around the Sylvian fissure connecting the frontal and temporal lobes. Now, the next step would be to remove the frontal parietal and temporal operculum, remove the insula to expose the external capsule, and then we also cut the arcuate fasciculus to expose the association white matter underneath. And this is the result that you will get. So now we expose a lot more short and long range association fibers. You can see from the top, if we go through them, you see the U-shaped fibers, again, connecting neighboring gyri. Then you see the SLF, or superior longitudinal fasciculus, which is a frontal parietal connection. The next one is the arcuate, and you just see it arching towards the temporal, but we cut the fiber to expose the white matter underneath. And then suddenly it gets really busy on the slide. So the first one is the IC, the internal capsule, or corona radiata at that level of the brain as well. To the left of it, you see that the tissue changes. So this is actually the putamen that you can begin to see and appreciate here. Underneath the putamen, you have the external capsule, and that is made up of the uncinate fasciculus at the ventral part of it, and the IVOF running on top of it and connecting the frontal and the occipital lobe. If we direct our view towards the occipital lobe, you can also see that there is the VOF, as it's often called, the vertical occipital fasciculus or system emerging here. And you can nicely see that the IVOF fibers have a perpendicular orientation to the vertical occipital fasciculus. Now, if we continue on, so now we're coming from the medial lateral surface of the brain. And again, for orientation here, we see nicely the corpus callosum, which is the landmark structure to identify here. Then underneath, you see the white matter of the fornix arching. And once we have the corpus callosum, we know that on top of the corpus callosum runs the cingulate gyrus. Now, the dissection on the medial surface of the brain usually begin within the cingulate sulcus. So you start removing the gray matter at the depth of the cingulate sulcus, and this is the image that you then end up with. So you can nicely now see the streamlines, the fibers of the cingulum that is running within the cingulate gyrus and arching into the temporal lobe here. Now, the cingulum extends from the subcallosal area in the anterior part of the brain, arches around the corpus callosum posteriorly on the dorsal surface, and then runs around the splenium to dive deeper into the white matter of the hippocampal gyrus. Now here, for orientation, I flipped the brain. So now we're looking from the top of the brain, or the dorsal view. And I did this to show you the radiating fibers of the corpus callosum. So especially in neuroimaging, when you're used to T1 sagittal views, for example, you always see that nice C-shaped corpus callosum, but it's really hard to appreciate how beautiful the corpus callosum is radiating into all the lobes except for the temporal pole, where the anterior commissure is the commissure connection. And it really, when you look at it from the top with both hemispheres, it kind of looks like a beautiful butterfly with the wings going up and down. It's a really, really lovely structure to dissect. But actually getting the individual radiations is rather difficult. So I wanted to show these to you. Then in homage to yesterday, I also threw this one in. This is not particular or per se white matter as such, but I wanted to show you the hippocampal structure because that came up a lot yesterday. And I always knew that it was called hippocampus from Greek seahorse, and I saw it in textbooks, but I was absolutely stunned the first time I could see it in the actual brain. It's such a beautiful structure. And if you're still struggling to see it, let's see, yes, the animation is working. It's right here. So this is where you can find the hippocampus. So this is a very quick run through of 2 to 3 days of Klingler postmortem dissections to give you an idea of what you can do with the method. And to summarize, as I said, it's invasive by nature, so you cannot under any circumstances do this in a living the human brain. It is time consuming, and both in terms of the preparation that goes into it, as we saw in the beginning. Some people freeze their brain for up to 2 months or even longer. So you have to allow for that preparation time, but then also the dissection does take quite a bit of time. In terms of performing the dissection, I always thought I knew my white matter anatomy until I actually tried to dissect it in the postmortem brain, and all the beautiful colors were taken away and the references where to place your regions of interest. You learn anatomy deeply when you do those dissections, and it's absolutely fascinating. And it really helps with the 3D understanding of where things are and how they connect to each other. As you've seen, in order to go from the brain to the dissection, you have to peel away the cortex. So by definition and by virtue of the method, it becomes very difficult to determine the cortical terminations of the white matter. The other argument is that with the process of freezing and thawing the brain, you might actually artificially create artifacts by separating the tracts that shouldn't be separated or pushing together others that may not necessarily be together naturally. And that is because obviously there is water in the brain. And what happens when you freeze water, it turns into ice, and ice needs more space than liquid water. And thereby you might actually artificially introduce some artifacts. That said, it is an excellent complementary research tool for neuroanatomical studies. It was for a very long time considered the gold standard for tractography, but as I've just shown you, the limitations are comparable actually. So it's still a very valid method, and it's unparalleled in the way we can understand the 3D structures in the brain. But it does have its limitations, and it's important to be aware of them. So that's the one method that we have available to us. The other is diffusion weighted imaging, and you've already seen this animation from Michel. So we basically just peel away the cortex again, but this time virtually, to look at the connectional anatomy underneath. Now, what do you need to do diffusion weighted imaging tractography? You need diffusion weighted data. And for the physicist amongst you, don't freak out. I know I'm wearing a watch and metal on me in that picture. It's a mock scanner, so no one was harmed. And you do need software and know-how, which is what we're going to give you today. Now, if you put those two together, you can get beautiful, beautiful images like this. And I stole this from Alex Lehmans, who nicely put that on Twitter. And what you see here is the arcuate fasciculus arching around. You can appreciate the IFF behind it. You can see the cortical spinal tract going down. And I just want you to lean back and relax for, I think it's 2 minutes. And I try to give you some anatomical pointers throughout. So here you now see the brain from the front. We're coming around on the left surface. You see the arcuate fibers arching. You see the uncinate that's connecting the anterior temporal and the inferior frontal. We're zooming in into the inferior parietal area of the arcuate connections now. One thing that is important to know here is that even though it looks like beautiful axonal structures, we're not actually seeing individual axons. And we're going to have a talk this afternoon about the details of the methods. But what we're seeing here is the streamlines, not individual axons, because the resolution is too big. So now we're flying anterior posteriorly over the corpus callosum, which is the red connections, commissure, and we see the cingulum on either side lying atop of the corpus callosum, mainly in green and purple here. So now we're looking forward, but we're going backwards towards the splenium, towards the back of the brain. You see the callosal radiations coming up. You nicely see the cingulum now arching around the splenium, diving into the temporal lobe. The cerebellum just came up, and now we see the corticospinal tract going down into the brainstem. We swing around, we look at the front of the brainstem. We can see the pontine fibers here, nicely going around the brainstem. And zooming out again to see the lateral surface again. Here we go. Now you see the occipital projections of the IFOF. You see the connections of the cerebellum. Voila. I think it's an absolutely stunning animation that Alex did there. Right, so tractography allows us, as you've already seen today, to dissect the white matter of the brain in the living human brain. And according to the classification, we have association pathways that connect regions within the same hemisphere or intrahemispheric connections. They're classically shown in green. We have projection pathways that have a superior-inferior orientation. They're classically shown in blue. And then we have the commissure connections that interconnect the two hemispheres, and you see them usually shown in red, and you see the corpus callosum here. Now, with that method, we can also dissect individual tracts, and there is pretty much no limit to what you can do if you have the right data available to you. So, brief summary, diffusion tractography is the only method that we have to look at the white matter in vivo in the living human brain. It is also time consuming, especially if you want the most stunning tractography like we've just seen in the video. It does take quite a while to acquire the data, process the data, and visualize the data. But the more you do it, the faster you become. That's one of the advantages. Again, you need to know your white matter anatomy or use it as a tool to learn your white matter anatomy. It is difficult to determine the cortical terminations because, especially with DTI, which is the most commonly used tracking algorithm, especially for clinicians, you cannot get too close to the cortex because the tracking won't work anymore. But as we've nicely seen yesterday in Christian Vollmer's talk, you can now do a lot with multimodal integration of images and have a very good estimate of where that white matter is going to. And also there's a big methodological push to do tracking close to the cortex or even into the cortex. Again, depending on the tracking algorithms we use, we might be prone to produce artifacts, and I'm going to show you a lot of examples of that later on. Tractography overall is an excellent research tool for neuroanatomical studies and a wonderful tool to learn your 3D anatomy. So with that, I don't want to keep you waiting. So now it's your turn. We're going to have a 15-minute break for those of you who didn't download the data yet or didn't install the software yet. If you could do so, Michel and I will be around for tech support where needed. And I see you in 15 minutes for your first dissections. Well, give us a shout if you need help. If not, we're just going to leave you with beautiful music and see you in 15. Thank you.